This program is a presentation of UCTV for educational and non-commercial use only. Check out our YouTube original channel, UCTV Prime, at youtube.com/UCTV Prime. Subscribe today to get new programs every week.
The aging but resilient brain, keeping neurons happy. First, some disclosures. Most of my support comes from the National Institute of Health and various grants. We also have some private foundation support and a little bit of royalty from a test publisher. And most important are some of the acknowledgments. Speakers typically wait till the end for the acknowledgments. I'd like to start with them because I think they're so important. And the first set of acknowledgments goes to the entire Memory and Aging Center, which is really a wonderful multidisciplinary group that has a very active clinic, a very active research program with probably 125 staff members at this point, and growing, forever growing. And this is a picture of us at our last retreat. I'm sure you could find Lisa in there somewhere. And I also want to acknowledge some very specific groups within the Memory and Aging Center. First of all, this is our aging team. And anyone who is, I'm the only one who's aging. The other ones are looking great. And if there's anyone here who's been part of the Memory and Aging Center aging program, you're familiar with Diana, Alex, or Jessica. They do a phenomenal job. And also some acknowledgments are in order for the neuropsychology postdoctoral fellows, in particular Bree Betcher, who does a lot of the work on neural inflammation that I'll be mentioning today, and Christine Welsh, who does a lot of sleep research, some of which I'll be referring to today. So again, can't do anything without them. So a quick outline of what I'd like to accomplish today. One is just to review some basics about the brain as they pertain to brain health and aging. I wanted to talk a little bit about some of the direct and indirect effects of aging on brain structure and function. And the reason why I want to spend some time on some of the not so good things that happen with aging is because once we understand these more clearly, we can start thinking about how to effectively target or design interventions so we can think of some steps we can take to then mitigate age-related problems. All right, so let me first introduce the neuron. As you can see, it's a happy neuron. And I want to go over just some real basics, and I think the relevance of all this will come into play when we talk a little bit more about some of the aging-related changes. So this is the cell body. These extensions here, these branch-like items, are called dendrites, and at the tips of these dendritic spines are the points of contact that this particular neuron has with other neurons. So these are the points at which it's receiving information from dozens, hundreds, thousands of other neurons. This long extension here is the axon, and these extensions here of the axon are the points at which this neuron is communicating with other neurons down the road. So that's basic neural structure. The way in which the neuron communicates is through these biochemical electrical impulses that travel down the extent of the axon. And at some point, evolution figured out that to have these nerve impulses propagate down the whole length of the axon, and I should point out that some of these axons are incredibly long. They could go from the top of your head all the way into your spine. So they can be some very long axons. Evolution figured out that it's a much more effective way to communicate information or propagate these impulses if it skips rather than rolls. And the way the neuron has evolved to allow the skipping to happen is these little myelin sheaths. These are fatty substances that other cells create, and they wrap around the axon. And this dramatically improves the efficiency of how well our neurons can communicate with another. This will become relevant in just a minute. All right, so this is a slice through the brain. It's as if you're looking at someone's brain and it was sliced pretty much down the middle. And there are a couple of things that you can see right away. The first is that there's gray matter and there's white matter. The gray matter are basically dense collections of cell bodies. And as you can see, the surrounding surface of the brain, referred to as the cortex, which is Latin for bark, it's the covering of the brain, is all gray matter. And it's the cortex that has evolved so dramatically over the last, umpteen millions of years. And it's the densest and richness of our cortex that makes humans so capable of so many things and enriches our cognitive capacity. Then you've got a lot of white matter. The white matter represents these axons that, as I mentioned, are covered with this fatty substance. So on a slice like this, they are looking white. And in some imaging approaches, these areas of white matter will also look white depending on the kind of MRI. And I don't know if anyone here has seen an MRI or taken part in an MRI experiment or will if they wind up working with Lisa, but that's what some images are going to look like. All right, so with that in mind, let's go over some basic brain facts. So there are about 80 to 100 billion of these neurons tucked away inside your skull. In addition to that, there are about just as many glial cells. And the glial cells, again, the glue that surrounds all these neurons and helps sort of maintain the structure. One of the types of glial cells are the oligodendrocytes. These are the cells that make the myelin, the fatty substance that wraps around the axon. So it's all fairly connected in there. These 100 or so billion neurons, again, are very richly connected to lots and lots of other neurons to the point where there are probably around 100 trillion synapses. The synapses are the connections between various neurons. So it's quite the computer. So, pretty good system. Let's review a few more brain-related factoids. First of all, the period in life during which you are going to have the most number of neurons is during your last trimester as a fetus. Okay? It's downhill from there. Right? And it's historically been assumed that we don't make new neurons, that what we've got now is what we started with however many years ago. So neurons are not being formed. Myelin. So myelin is something that these oligodendrocytes makes very, very slowly, and it's not like it's replenishing old myelin. The old myelin is still kind of there, and myelin ages, it starts to shear, starts to split, kind of like, dry hair, you get split ends, and it doesn't work so well. In addition to that, the brain is what I would refer to as a high-maintenance organ. So it takes 15% of the cardiac output goes to your brain. It only weighs a few pounds, but it's taken 15% of the cardiac output and about 20% of your total body's oxygen consumption. In addition to that, it's a very, very picky eater, whereas most cells in our body will take glucose, they'll metabolize lipids, fats. The brain will only take glucose, and it consumes about 25% of the total body glucose. So, with engineering like this, with cells that don't reproduce, and myelin that gets old, and a high-maintenance system that requires lots of glucose and oxygen, what could possibly go wrong? Everything, of course. So let's look a little bit about what happens with typical aging and brain structure. I mentioned earlier that the cortex is where it's really happening cognitively in humans. So one of the things we can look at is cortical volume. This is a schematic of the brain with the various lobes. And we have measured cortical volume in our subjects. And this is, again, data from the Memory and Aging Center with Again, community dwelling, high functioning, typically aging individuals from 50 to 90. And although you can see a fair amount of scatter in the total cortical volumes, with some people being a little higher, some people being a little bit lower, but the general trend with aging is linear and downward. So there is some, not necessarily cell loss, but shrinkage of the cortex with age. We also see some changes in that white matter I was referring to. Again, lots of scatter around the regression line, but the regression line still tells the tale, which is that as we age, our total amount of white matter is decreasing. So could this be irrelevant? It could just be some brain shrinkage, but size doesn't matter. It's all about function. So do we see any age-related changes in brain function? Well, I think that the thing that people complain about most as they age is, well, I don't remember as well as I used to. So we can actually measure age-related changes in memory. This is a 16-word list that we give to actually a lot of our subjects, and we give them many opportunities to learn the list, and then 20 minutes later we say, tell me all the words on that list. And this is what happens across the adult age spectrum. So again, a fairly linear and downward trend with age. Well, memory is not the only thing that changes with age. We also tend to get a little bit slower. So these are some computerized processing speed tasks. They don't require a lot of cognitive ability, they just require speed, and this is what happens with speed as we get older. The higher you are, the slower you are in this particular graph. So as you can see, there's a general steady slowing with increasing age. Novel problem solving. Here's an example of a kind of task that might be used to challenge someone's spatial reasoning. So there would be, say, a matrix like this, and you have to decide which of these 6 designs here most logically fits into this empty box here. And this one is something that I was able to figure out, so I'll share the answer with you. But you have triangles here that move over to squares, and as it goes down, you have one black bar, 2 black stripes. So we probably want a square with 2 stripes in here. So I would guess number 3. This one took me a lot longer. Then I won't tell you the answer. But these are the kinds of tasks that are administered to tap into things like novel problem solving, and this is what happens with age. So that even starting in our 30s, so for those of you who are under the age of 50 who are starting to feel smug, let me really point out that some of these age-related cognitive changes occur early on. And again, you see a steady change with increasing age. I should emphasize that there are some things that don't decline with age, and things like vocabulary, general fund of verbal information, and my bias is that, of course, wisdom increases with age. But I think it would be good to start thinking about a model that will help us understand all the various components that contribute to changes in brain structure and function. So at the end point here, we have cognition, memory, judgment, reasoning, problem solving. That's obviously influenced by brain structure and brain function. So as we age, what are some of the things that are happening? Well, a lot of things that are happening that can influence brain structure and functioning. I put really just a subset of some of the age-related components that influence brain-related. And I think one of the things I should really emphasize with this particular graph is that whereas all of these potential influences on brain structure and functioning are listed individually, in reality, they're all very richly and deeply interconnected. So there should be dozens of lines connecting all of these highly interactive components that are influencing how our brain changes with age. But let's start reviewing some of these in the hopes that as we better understand these, we can think about how we can work around them, mitigate them, avoid them altogether, compensate for them. One note before we get started. So when you review the literature, what you notice is that there are really a couple different kinds of questions that researchers are asking. So some people are really asking the question, what sorts of life changes or variables are associated with dementia, and what are some of the things that reduce our risk of dementia. Whereas other researchers are saying, we're really interested in identifying things that improve or stabilize cognitive functioning as we age. And these are very, very different questions. There are lots of things that potentially reduce the risk of dementia that have no impact on cognitive functioning if you're neurologically intact. And there may be lots of things that improve day-to-day cognitive functioning but won't make a dent in a neurodegenerative condition like Alzheimer's disease. But it's important to keep in mind that when I describe some of the studies, they're asking different kinds of questions. The other thing to point out is that there are really a couple different kinds of research designs in these studies. The first are the epidemiological or observational studies, and a lot of what we do at the Memory and Aging Center would be an observational study. The one that Lisa described is an observational study. And the important thing to emphasize here is that we can identify, and I'll be talking about, clear correlations between, say, memory and some age-related change in brain functioning. But that doesn't mean that it's a causal relationship. All we have been able to demonstrate is that there's an association. We'd like to imply causality, but we really can't. In order to really get at causal mechanisms, what we need to do is carry out clinical trials or very specific experiments. Now, these sorts of observational studies are easier to do. They're easier to recruit for. You can get much larger sample sizes, but again, at the cost of not being able to confidently assert causality. These experiments and trials are much more expensive, much more difficult to do. You rarely have large samples. And then less statistical power, but you can make more inferences about causality. All right, so with that in mind, let's review some of the things that negatively impact brain structure and functioning. And I really need to start off with protein aggregation, because we can't really have a talk about keeping neurons happy without talking about dementing disorders. And protein aggregation is really what happens with these neurodegenerative diseases. These diseases all have this insidious onset, gradual progression, and they're all associated with an abnormal accumulation of some kind of protein. And I'll say more about that in a minute. And they all selectively target different brain regions. So the most common neurodegenerative disease to all of us, of course, is Alzheimer's disease. And there are a couple of different abnormal proteins that are clearly related to the mechanics of the disease. And one of the proteins is amyloid, another one is tau. Parkinson's disease is caused by abnormal accumulation of a protein called alpha-synuclein. There are other proteins that also cause different sorts of disorders, all of which have as an endpoint significant deficits in cognition and behavior. Alzheimer's disease, of course, is the one on most people's minds, in large part because it's clearly a disease of aging, and the incidence of Alzheimer's disease, unless we can figure out what we can do about it in the next 20, 30 years, is going to really start increasing exponentially to the point where there will probably be close to 1 million cases, new cases a year by 2050. And the pathology of Alzheimer's disease, I mentioned that there's this abnormal accumulation of misfolded amyloid, which is seen under the microscope. But these are extracellular accumulations called plaques. There's also intraneuronal misfolding of a protein called tau that creates these tangles. But I'll mostly be talking about the amyloid plaques as we go forward. And I just want to demonstrate this for a moment because, again, it's going to be relevant later on, because anything that can change the rate at which these proteins misfold is going to have a huge impact on cognitive functioning in, in potentially millions of people. So here's what happens with these proteins. I mean, why is it that these proteins are misfolding and causing these horrible diseases? So this is an example of an amyloid precursor protein, which is a normal protein. It does its job in the synapse. It helps maintain synaptic health, and they're all kind of very squiggly like that. And then, over time, what our bodies are always doing is they're chopping these proteins up into little bits, reabsorbing them, rebuilding them, and putting them back out there. So there's a constant creation of new proteins. And what happens in Alzheimer's disease is that these proteins are getting cut at the wrong spot. And because of the way in which these proteins then become misshapen, they start to glom together. And once they start to glom together, they form these plaques. And whether it's the free-floating abnormal proteins or the collection of these abnormal proteins that are causing dysfunction is not altogether clear, but there's no doubt that beta amyloid is playing a role. So anything, of course, that either inhibits development of these abnormal proteins or helps our brains get rid of these abnormal proteins can reduce the incidence of not only frank dementia, but even subtle cognitive impairments that occur with aging. So that's protein aggregation or neurodegeneration, and there'll be some references later on to that as well. So another thing we need to really take into consideration is vascular risk. And this is something that we can potentially do the most about. So what are some of the vascular risk factors? Hypertension, high cholesterol, high lipids, diabetes, or even hyperglycemic states. Obesity, again, a huge vascular risk factor. Smoking, huge no-no. And just getting older is a vascular risk. I mean, it is. I mean, it's, even if you do a great job of holding all of this stuff steady, as we age, there is increasing risk for cerebrovascular disease. And this is one of the ways in which cerebrovascular disease can present. So this is an MRI scan, and you can see these tiny little holes in sort of the middle part of the brain. These are just tiny, tiny strokes or lacunes. And they're frequently asymptomatic. If you take 100 70-year-olds who are just perfectly fine and you put them in the scanner, about 15 to 20 will have evidence of having a small stroke that they never even were aware of. Some of our research suggests that these silent strokes aren't really silent, but do have a subtle impact on cognition. And then another ways in which cerebrovascular disease can present is with this abnormal signal in the white matter. So there are probably ischemic changes, insufficient blood supply to the white matter, and you get scans that look like this. And it's very clear that not only do these white matter signal hyperintensities increase with age, and this is a large sample of the community dwelling elderly in the Framingham study, but an increase in white matter signal hyperintensity is directly associated with a decline in executive functioning. And by that I mean multitasking and novel problem solving and inhibition. So what we did is we took 50 very healthy community dwelling elderly, imaged them and tested them at baseline, and then we followed them for 4 years, and 4 years later we tested them again and looked at their scans. And what we found is that you could divide the group into quartiles based on what the increase in their white matter signal hyperintensity was. So the people with the least amount of increase in white matter signal hyperintensity actually had like a 3-point increase on their executive scores. Now, so basically no real change. The people in the second quartile, so they had a little bit more white matter signal hyperintensity increase. Again, didn't really show much change. Third quartile didn't really show much change. But the 25% that had the most increase in white matter signal hyperintensity were the ones that showed the more significant drop in their executive functioning. And again, I should emphasize that even after 4 years, they were still normal, healthy, community-dwelling individuals. We've also been able to show that this atherosclerosis is associated with change in executive functioning. So one of the ways you can measure atherosclerosis is looking at the thickness of the walls around the carotid arteries, and you can do this with ultrasound. So we measured carotid intimal wall thickness in several community-dwelling elderly, and then we measured their cognitive functioning at baseline and again about a year or two later. And we noted that as they had more evidence for atherosclerosis, as the thicker their walls of the carotid were at baseline, the greater the drop in executive functioning over that 1 to 2 year period. So lots of evidence to suggest that vascular risk factors are not good for brain functioning. And then I think some very intriguing data that has come out of the Northern California Kaiser system. So Christine Yaffe, who's a colleague here, and Rachel Whitmer, who's at Kaiser have done a couple of very interesting studies. So in the 70s, Kaiser did these very extensive multimodal assessments of health in a large number of subjects. And then, of course, these people stayed in Kaiser, and we now can look at their data 36 years later. So at middle age, when they were middle age, when they first went into this study, those who were obese, they had the highest body mass index. This was associated with a threefold increase in the risk of Alzheimer's disease. They were much more likely 3.5 decades later to come in with Alzheimer's disease, and fivefold increase in Alzheimer's, in vascular dementia. And then they went ahead and looked at the kind of obesity, because there are different kinds of obesity, right? You could be apple-shaped and you could be pear-shaped. Apple-shaped is when you have a lot of the belly fat. Pear-shaped is when it's more in the hips and butt. And they basically, and there's been some theories out that says that central adiposity, the belly fat, is the worst for you. And in fact, they found that the people who had the most amount of central obesity had a threefold increase in a diagnosis of dementia, again, 36 years later. So, what we do in midlife has a clear impact on the evolution of our brain structure and function. Getting back to our model, neuroinflammation. And this is some of the work that Bree Betcher is doing. The inflammation story, I think, is very intriguing. So if you hurt yourself, you mount an inflammatory response. That's healthy. That's what you should do. We now know, however, that when there's a peripheral injury, like you burn your arm, for example, not only is there inflammation around the arm, but there's a fairly rapid inflammatory response that occurs in your brain, which is okay too. That's also adaptive. And when you're young, you have a spike in inflammation, which then subsides. When we're older, it doesn't subside as quickly, so we have more sustained neural inflammation. And this sustained neural inflammation is particularly evident in a structure called the hippocampus. And the hippocampus is tucked away in the middle of our temporal lobes. And one of the things that the hippocampus does is enables us to lay down new memory traces. So if you have lesions in the hippocampus, you'll be amnestic. You're not going to be able to learn and remember new information. Why is the hippocampus so important for our discussion? Well, this is ground zero for Alzheimer's disease, which is why Alzheimer's patients have very significant memory problems. So it seems that, at least in laboratory animals, neuroinflammation is associated with hippocampal and memory problems. So we looked at that in humans. So we had 141 subjects, again, as part of our normal aging studies, some of whom had detectable levels of a protein called C-reactive protein, which is associated with ongoing inflammation. So if you're chronically inflamed, you'll have some elevations in your C-reactive protein. So we had 76 subjects who had some detectable CRP in their blood, 65 of whom did not. We looked at their memory performance to see if, in fact, the nature of their memory performance made us worry about their hippocampi. So again, I should emphasize that all of their memory performances were average. None of these were not impaired people. But we looked at how well they remembered a list of words immediately, and there was really no difference between the group that had detectable CRP and those who didn't. But then we looked in to see what happened after 20 minutes. So we were measuring how well they could consolidate or lay down a new memory trace. And this is where you got a very clear difference between the group with detectable levels of CRP. They did show some greater difficulties, relatively greater difficulties in learning new information. And when we looked at their scans, lo and behold, they also had smaller medial temporal lobes. Another way of looking at inflammation, and there are other inflammatory markers that we're now studying, including TNF-alpha, IL-6, and we wanted to look at in a more detailed way what's happening to the white matter in the brain. So there's fancy imaging techniques called diffusion tensor imaging that can give you an idea of what the microscopic integrity of the white matter is. So even if it looks normal on a regular scan, we can get a better measure of the microscopic integrity. And what we find the area of white matter we were looking at is in green here. And we saw a clear association between these inflammatory markers in blood and the amount of, or the integrity of the white matter in certain fiber tracts. So again, inflammation, neural inflammation, if chronic, can have some lasting impact on brain structure and function. Again, going back to our model, sleep. So this is sleep over the lifespan. So we've got, the first few days of life and first few months of life here, going all the way up into the 80s. And you can, this is the total amount of sleep that we're getting typically. You can notice here that babies pretty much sleep like babies. And there's a steady decline in the average amount of sleep we get as we get older. In addition to that, the kind of sleep we're getting changes, so that the amount of REM sleep gets smaller and smaller. And there are lots of very interesting studies that talk about the potential importance of REM sleep for our ability to consolidate new information over time. So again, this could also be affecting cognition. So let's look at sleep in a 34-year-old male, and this is done. We hooked the subject up to these electrodes and had recordings overnight. And in red is when they were awake, and pretty much is when they went to bed. And you can see these are sort of 5-minute epochs. And the first person fell asleep pretty quickly, went into light sleep, went into deep sleep, went into some REM, more deep, light sleep, deep sleep. As the evening wore on, a little bit more REM, which is normal. And at the end of the day, and came morning, woke up. Now, let me take some questions at the end. That's all right?
Just real quick, what is the definition for REM sleep?
Rapid eye movement. So there's, if you look at an EEG, there's greater activity, and your eyes are moving around, which is why it's called REM, rapid eye movement. This is a 68-year-old who, as you can see, took longer to fall asleep, went into light sleep, deep sleep, REM, woke up, fell back asleep, woke up, light sleep, REM, woke up. I suppose if we graphed the bladder, it probably would be somewhat similar. But you notice that there are a lot of awakenings, and this is not at all atypical with age. And in fact, the amount of REM sleep did not increase as the night progressed. So this is an abnormal sleep pattern, but for a young person, but not typically, or not abnormal for an older person. What happens, and of course, sleep is obviously very important. You all remember from all-nighters that 24 hours of sleep deprivation is not good for your cognition. But what happens if there's an habitual reduction in sleep? So people reporting poor sleep tend to perform less well on memory and executive tasks. And as, again, normal older folks, as their sleep disturbance increases, the risk of cognitive impairment increases. Insomnia, which is either difficulty falling asleep or difficulty staying awake. So cohorts of insomniacs have been studied, and they have a whole range of cognitive difficulties, relative cognitive difficulties, compared to people their age who are sleeping all right. One of the things that, one of the reasons why we're so interested in sleep at the Memory and Aging Center is that studies with transgenic mice. So these are mice who are prone to Alzheimer's disease, and if you restrict their sleep, what you notice is that there's an increase in this amyloid plaque deposition. So the biology of Alzheimer's disease may occur a little bit more readily during, at least in laboratory mice, associated with poor sleep. All right, so I'm not going to say much about genetics, in part because there's not a whole lot we can do about that, although I should mention there's some really interesting studies coming down that talk about how we can potentially influence gene regulation. So genes are turned on and turned off all the time, and all the genes are doing is producing messenger RNA that's going to downstream turn into a protein. So if there's some ways that we can manipulate the rate at which these genes are turned on and off, we can potentially regulate protein production, which, as we know, could have some impact on these abnormal protein aggregation that causes neurodegenerative disease. And I'm not going to say much about lifestyle, because that's really all we're talking about, right? Lifestyle is how we're going to try and keep our neurons happy. All right, so to summarize so far, aging can clearly have a significant impact on brain structure and brain function, but to emphasize here that these changes are neither universal nor are they inevitable. There's some people who used to say that if you lived long enough, you'd get Alzheimer's disease. I don't think that's true. And in fact, even though I showed you lots of graphs that showed how cognition declines with age, I have other graphs in my repertoire that show that, well, if you follow the same individual longitudinally over several years, that you can demonstrate that a large percentage of healthy elderly are holding steady. So I think the question here is not whether or not whether we change or decline with age, but what are the factors that make it more likely that we're going to hold steady? In other words, keeping those neurons happy. Multiple factors, and they're all very interactive, influence how the brain changes with age, and we're going to talk about the ones that are potentially under our control, and certainly that's one of the huge goals we have at the Memory and Aging Center. This is what we're targeting. So, what can we do? And I'm going to briefly run through some of the items here, and this includes starting off smart, physical exercise, cognitive activity, social activity, sleep, diet, medication, supplement. So I'm going to try and touch on all these areas, although there's some of these areas that we know more about and some that we know less about. And I will try and convey when I'm not very confident about what it is that I'm saying. So, start off smart. That's on the top of the list. What do I mean by that? Well, I mentioned that amyloid is the protein that accumulates in Alzheimer's disease. And about 6-7 years ago, some folks at Pittsburgh came up with an imaging compound that binds to amyloid in the brain. So then you can use a PET scan to take pictures of people's brains, and you can measure how much amyloid there is. And here's a patient with Alzheimer's disease, and all the red is lots of amyloid, and the yellow is a little bit of amyloid. And here's an age-matched, cognitively controlled elderly, and you can see that there's, maybe a tiny bit of PIB binding, of amyloid in the white matter, which is just an artifact, doesn't mean anything at all. In other words, you don't see any amyloid in this particular individual, right? So all is good. Amyloid separates Alzheimer's disease from normal elderly. All right, take a look at this person here. This is a cognitively normal individual. And look at how much amyloid is in this particular person's brain. And if you take a sample of individuals who've died and gone on to autopsy, you'll find that a large percentage, 15–20% of people who were perfectly normal at the time of their death have, under the microscope, the changes that we associated with Alzheimer's disease. So it's pretty clear that there are lots of people walking around with pathological changes of Alzheimer's disease in their brain. That doesn't seem to be affecting them. And there are lots of people walking around with maybe some tiny little strokes. And there are lots of people walking around who've had a head injury 30 years ago that are doing much better than someone else who had a similar head injury 30 years ago. So what's explaining this? How is it that some people seem to be relatively immune to these brain injuries? So the whole idea of starting off smart refers to this notion of cognitive reserve. And cognitive reserve basically is a way of trying to explain how it is that some people are fairly resilient to pathological changes to the brain. And this is a phenomenon not just specific to the brain. I think that people who study spines have always been amazed at how someone will be scanned incidentally or go to autopsy, and they'll see tons of pathology in this person's lower back. And the person never had any lower back problems. So again, this is a phenomenon not just specific to the brain. So this has been studied fairly extensively over the last 10 to 15 years. And I want to mention the nun study since I think it's particularly intriguing. So almost 700 members of this particular nun order were agreed to participate starting in 1991. They got annual cognitive assessments. They got lots of blood work and neuro exams and cognitive assessments every year, scans, and then they all agreed to autopsy. So they all got autopsies when they passed away. And it's sort of this, I don't think this was originally intended, but it turned out that there was lots of archival material because these people had been in the convent all their lives. And one of the things that they had all done when they entered the convent was they all had to write an autobiography, and all this stuff was available. So here are some of the things that they found. Well, first of all, they found that when some of these nuns who were cognitively normal until the day of their death, they went to autopsy and they had a lot of Alzheimer's disease in their brain. So what's going on here? So they went back and they looked at these autobiographies that were written when they were in their early 20s and measured what they referred to as idea density. This is the sort of the richness of the prose or the writing style, the quality of their prepositions, the additional clauses, the basically noun and verb usage and vocabulary, and again came up with a measure of idea density. And what they found is that those nuns whose autobiographies 50 years earlier reflected a high degree of idea density were doing much better cognitively than their peers who did not start off with this high degree of idea density. And in addition, when they passed away, if there was Alzheimer's disease present, the ones who had the sort of the richer idea density at baseline were much more likely to have not shown clinical symptoms or had less prevalent or less severe clinical symptoms while they were alive. So something about how they were initially, and we can refer to this as intelligence if you like, but there's, and education is another marker of this cognitive reserve. So, you know, we think of this cognitive reserve as a way of accounting for some of these huge inter-individual differences in how vulnerable we are to you know, various injuries to the brain. All right. And you know, another way of looking at this is that if you have two people, one with high reserve, one with low reserve, that as the, say with Alzheimer's disease or strokes or a head injury, when that starts to happen, the person with low reserve will start to show decline. The person with higher reserve will maintain that high level of functioning for 1, 2, 3 years, 4 years before they have what they refer to as their point of inflection or start to decline. Well, cognitive reserve is great for you guys, but how about the rest of us who have to just, you know, rely on other ways of optimizing brain functioning? So with this in mind, I just want to refer to Marion Diamond and her colleagues. Marion Diamond was a pathologist at the University of California at Berkeley, and some of her colleagues, including Rosenzweig, who was in Southern California, and they were married, and they stayed like 450 miles apart throughout their entire academic careers. She has awesome cognitive reserve. She is now in her 80s, and I've heard her talk. She's a dynamo. But they were very interested in what influences brain development. So one of the things they did in the 60s was, and so if you were a rat in her lab, you would be randomly assigned to one of a few different living conditions. So this is the standard living condition. So you've got a small cage, you have a few rats in there to keep you company. There's the unlucky ones who are all by themselves in a cage with, again, nothing to play with. And then they had the enriched environments with, you know, exercise wheels and stimulating stuff all around and lots of other rats to hang out with, sort of like a rat Disneyland. And then they did a range of experiments, but of course at the end of all the experiments, the rats who had volunteered for the studies were sacrificed, their brains were looked at under the microscope, and here's what they found. So the rats that were in the enriched environments had increased cortical thickness, up to about 7% in some regions. So they weren't adding new cells, so there was no increase in the number of cells, but the neurons were bigger. They had more and longer dendrites, which meant that they were more able to communicate with other neurons. There were more glial cells, which are providing a lot of supportive environment for their functioning. The size of the capillaries were bigger, so the blood supply was better. And even so, if you were a young rat and you were in this enriched environment for just a few days, you could demonstrate some positive impact on brain structure. And what was most important is, at least for our purposes here, is that when they took older rats, even very, very old rats, and put them into the enriched environment for the first time, although it took more than a few days for there to be a positive brain effect, you got very significant improvements in brain structure over even a period of a month or two of being in an enriched environment. So that clearly demonstrates that plasticity is very active, at least in a rat brain, at any age. So what was it about these enriched environments, and what is it that we can then apply to our environments that's going to improve our neural functioning? Well, their enriched environments were fairly complex. They were certainly, the rats were more physically active. There was a running wheel and lots of stuff to play with. They were more cognitively enriched, and they were more socially enriched. And it'd be interesting to know which of these is the most important, or all of them. Hard to know. So let's talk a little bit about physical exercise in humans. So I'm going to start with a couple of epidemiological studies. So these are studies that look at thousands of individuals. They can do have them fill out questionnaires about their level of exercise, or in some cases questionnaires about their level of exercise 20 years ago. Not always the most reliable, because everybody overestimates how many times they were at the gym last week and how many minutes they were on the treadmill. But still, when you have several thousands of subjects, you can get away with a little bit of noise. And here's a great little study done by Christine Yaffe, where they had a large number of older subjects that were followed for a long period of time. But all she did was she asked them to indicate how much they walked. And they divided the group into 4 subgroups depending on how much they walked, and they measured changes in a cognitive test called the mini mental state exam. It's not a particularly sensitive test, so the fact that they got findings, I think, is very dramatic. And it's clear that the more these individuals walked, the smaller the longitudinal changes were in their test scores. So again, something as simple as how much do you walk is related to long-term cognitive outcome. It's also true that people who report a long history of being physically active are much less likely to show cognitive impairment in later life. And even if you wait until you're in your 50s or 60s to get more physically active, this too is associated with a slower rate of cognitive change, and even a lower risk of Alzheimer's disease. So this is a review of several studies. So someone did a meta-analysis where they took several different studies, combined all the data to see if they could find some distinct trends. So this meta-analysis included over 33,000 subjects. So it's a pretty powerful design, and subjects were followed up to 12 years. And the bottom line really is that a high level of physical exercise or physical activity was associated with a 38% reduced risk of cognitive decline. So one of the ways you can measure this reduced risk is something called a risk ratio, or an odds ratio, with an odds ratio of 1 being you're just as likely to have problems as anybody else. And the lines represent the range of confidence that the researchers had about their particular odds ratio. So it's always important to have these confidence intervals. But pretty much whenever you have the line that's not crossing 1, that you've got a study that supports the idea that physical exercise is associated with better cognitive outcomes. And as you can see, the vast majority of these studies demonstrated that. So I think that the epidemiological evidence linking exercise with reduced dementia and better cognitive outcome is very, very strong. And there's some direct effects on the brain. So people who exercise more are more likely to have greater brain volumes, particularly in the white matter and in the hippocampus, which again we know is so important for memory, and regions of the frontal lobes. There's better cerebral blood flow, better memory, better executive functioning. All of these are, at least with observational studies, associated with better cognitive outcome. Well, what are some of the mechanisms by which physical exercise could be improving our brain structure and function? Well, it's complicated. I think that as we exercise more, we start to lose some weight. So there's you're reducing your vascular risk factors. You're reducing inflammation. Well, even if you hurt yourself every once in a while doing the exercises, the long-range outcome for inflammation is a positive one. And then potentially some direct impact on neural function that I really want to talk about. So there's a protein called brain-derived neurotrophic factor. It's one of several proteins that are very active or involved in supporting the health and functioning of our neurons. And this BDNF, brain-derived neurotrophic factor, is very heavily expressed in the hippocampus and the cortex. Again, structures that we know are particularly important. And involved in neurogenesis. Neurogenesis basically refers to the ability to form new neurons. I mentioned that the historical belief is that we don't make new neurons, but I think over the last decade or so it's becoming increasingly clear that under some circumstances we can form new neurons, and the hippocampus seems to be one of those places where it's really happening a lot. So at this point, let's say you survived Marion Diamond's lab, you moved to Southern California, hoping to get into real estate, but you wound up just needing to get a job in Carl Kottman's lab. He's very interested in looking at the effects of physical exercise, and he does controlled experiments, and rats and mice are his preferred organism. So he wanted to look at physical exercise and its relationship to brain-derived neurotrophic factor. So he had some rats reflected up here who, and incidentally, if you put an exercise wheel in a rat's cage, most of the rats really enjoy it. You don't have to coerce them to get them to run in the wheel. They'll do it spontaneously, and some of them will just kind of run all night. So he had wheels, exercise wheels, for some of the rats, and nothing. The sedentary rats, of course, didn't have access to that. And he sacrificed them and looked at a marker of brain-derived neurotrophic factor in their hippocampus. And the brighter colors here reflect an increase in basically messenger RNA levels of the brain-derived neurotrophic factor. So again, direct evidence here that exercise is associated with more BDNF in the hippocampus. And in these very same rats, it was associated with improved memory. And even more importantly, I think, for our attempts to mitigate the effects of Alzheimer's disease, we have mice that are genetically engineered to get Alzheimer's disease. And in these mice, if they were exposed or given the opportunity to exercise early in life, they had fewer cognitive deficits as they aged. And even in older mice that were on these running wheels for as little time as a few weeks, he was able to document that there's some improved memory performance, whereas the mice who were not exercising showed a decline in cognition. So I think that the evidence for exercise is pretty overwhelming. And then to add one other highlight here, so Art Kramer, no relation, has a lab where he's been studying physical exercise for quite some time in individuals of all ages. So they did a randomized study with 120 older adults. It was a one-year study where subjects were randomized either to an aerobic condition or a stretching and toning condition. And then he looked at brain structure and function at the end of that one-year period. And the group that had the aerobic exercise had an increase in hippocampal volumes and an improvement in their spatial memory. In addition to that, he did serum measurements of brain-derived neurotrophic factor, and the group who was doing the aerobic exercise had higher levels of serum BDNF. So, how many of you are going to be at the gym tomorrow? All right, good. What about cognitive exercise? All this stuff about crossword puzzles or Sudoku, so whatever that is. I've never had the patience for it. And how many do you read books, visit museums, spend your evenings at UCSF and mini medical school, for God's sake? So there are lots of leisure activities that are promoting cognitive exercise, and there are also some very specific cognitive interventions, many of which you've probably been exposed to on late night television, because there's some ads. So does cognitive exercise really work? So there are some epidemiological studies that suggests that the more active individuals are intellectually, it may be associated with a lower risk of Alzheimer's disease and cognitive impairment in general. And then what I thought was a fairly intriguing data set from our colleagues at Berkeley. This is a paper written by Susan Landau, where she took a lot of older individuals and measured, with that scanning technique I mentioned earlier, she measured the amyloid in their brains. So she had patients with Alzheimer's disease, she had young controls, and then she had her sample of normal elderly subjects. So the Alzheimer's patients had a lot of amyloid in their brains, not surprising. The young controls didn't have any amyloid in their brains, not too surprising. But then she divided her older sample based on their self-reported level of cognitive activity when they were younger. And there's formal questionnaires that allow you to do that. You basically, in various stages of your life, you indicate how much time you spent reading, how much time you spent doing in school, etc. And she found that the group that had the lowest amount of self-reported cognitive activity when they were younger had the most amount of amyloid deposition in their brains. And the group with the highest amount of cognitive activity, were looking a whole lot like the young controls. And this is an observational study, not a clinical trial, so we can't say for sure what the cause and effect relationships are, but again, very intriguing kinds of data. There was a trial done about 10 years ago called the ACTIVE trial, where they studied almost 3,000 individuals between 65 and 94, and they were assigned to either a no-contact control group, memory training, or reasoning training, or using computers to improve speed of processing. And what they found is that after 10 sessions, that the subjects who were involved in one of these trainings got better. There was no generalizability, so that the memory training group got better at memory, but they weren't faster. The reasoning group got better at reasoning, but their memory didn't get any better. But they did a follow-up study a few years after all this and reported that the improvements maintained themselves over several years, which again was very, very encouraging.
So this study took place in 98, not 1890, right?
That would be true. Good eye. How come you didn't spot that before I was up here? So Guy mentioned that there are lots of formal cognitive training programs out there. And I don't think we're at the point where we can say with any degree of confidence that they are better than attending mini medical school, or if one program is better than the other. We need a lot more randomized clinical trials. We don't know if any of these training programs are generalizable to a fuller range of cognitive abilities, or whether they just improve your ability to do the thing that you're being trained to do. I think there's a lot more important research to be done, and we really don't know if cognitive training is going to help very much if you already have the earliest signs of one of these neurodegenerative conditions. So a lot more work needs to be done there. We also don't know nearly enough about the importance of social engagement on maintaining optimal neural health. Again, there have been some observational studies, epidemiological studies that suggest that groups that were individuals who are very, very socially active have a lower risk of developing Alzheimer's disease. Whether or not, but again, cause and effect is really not clear. We need clinical trials. Sleep, not enough research has been done with sleep. We do know that in studies of patients with sleep apnea, that improvements in their sleep are associated with improvements in cognitive functioning. So that's encouraging. Whether or not there can be either some pharmacological or non-pharmacological interventions that improve the quality of sleep in older individuals, and whether this has downstream impact on brain structure or functioning is not really clear. Anti-inflammatory medications. I mentioned the importance of thinking about inflammation as a risk factor for cognitive decline. I will again mention some of the more epidemiological studies. Here's one of the first ones that got people very excited. So about 7,000 subjects in Holland were followed, and they found that a certain percentage of this 7,000 wound up with Alzheimer's disease down the road. And then they looked to see if their use of non-steroidal anti-inflammatories, the ibuprofen, the Advils, and et cetera, or even aspirin, had any impact on the risk of developing Alzheimer's disease. So the ones who had some short-term use of the NSAIDs didn't really show any change. They had the same risk for people of dementia as people who did not take these medicines. People with intermediate use had a lower risk of about 0.83, and people who were using the NSAIDs for a long time had a much reduced risk of developing Alzheimer's disease. So again, this was a very exciting observational study that got a lot of people thinking about whether or not non-steroidal anti-inflammatory use could have an impact on either avoiding dementia or improving cognitive functioning. Other epidemiological studies, here's one study where they combined lots of study looking at lifetime exposure to these non-steroidal anti-inflammatories, and again, you got a very significant drop in the risk of developing Alzheimer's disease over time. So again, very exciting. So the next thing that people did was went into clinical trials. So if you are worried that someone has Alzheimer's disease, or if they do have Alzheimer's disease, you can randomly assign them to an NSAID or a placebo condition and look to see if there are any differences between those groups. And this is one of those instances in which the NSAIDs as an intervention were not successful. Are they successful as a prevention or a delay of cognitive change? Possibly, but as a direct intervention, no. Diet, we really need to mention diet. Anything good for the heart is good for the brain. In the Mediterranean, there's fruits, vegetables, cut down on the fatty foods, increase the amount of fish. So I think there's several studies, epidemiological, that really suggest that these sorts of diets are not only good for your cardiovascular health, but are better for your brain. There's tons of supplements out there, and if you go to the health food store, there's a whole slew of great interestingly named supplements that are supposed to promote your brain health. Fish oil and resveratrol, which is it's in the grape skins, ginkgo biloba, the various vitamins. So the data in support of any of these as an intervention that's going to reduce risk of Alzheimer's disease, again, the data are simply not out there. They're not really very supportive, and data supporting the notion that these supplements might improve cognition in someone who doesn't have a disease, again, those data aren't really very compelling either. So to sum it all up, what does it take to keep neurons happy? Well, start off with good genes. That's important. Physical exercise, I would say absolutely that's going to be a very key component for maintaining cognitive health and staving off things like Alzheimer's. Cognitive activity, there's some data out there that are very promising, but we don't really know a whole lot about the specific kinds of cognitive activity or just how important that's going to be. Social activity, again, we don't know enough, but I would recommend being socially active. It's not going to hurt. Good sleep. If you took a nap during my talk, that's okay, because that's an important part of brain health, and that's what this is all about. Minimize inflammation. I think that the data out there are pretty compelling. Maintaining a great diet. You have supplements, not so sure. Low fat, maybe, again. There's not any great clinical trials out there. Copious amounts of red wine and dark chocolate. God, I hope so. And that's it. Thank you for your attention. And we'll have time for questions. So what's the relationship between diabetes and Alzheimer's? So diabetes is traditionally thought of as a vascular risk and associated with increased chances for stroke. But we now know that increased diabetes is associated with a greater rate of Alzheimer's disease. Whether or not it's due to the cerebrovascular disease, which makes you more vulnerable to showing symptoms with the same amount of Alzheimerization of the brain, or whether there's some specifics around inflammation and glucose, or insulin resistance. That's another theory that's been bandied about. And there's some suggestive data showing that poor glucose metabolism and insulin resistance might be part of this complicated neuronal cascade that leads to improper cleavage of beta amyloid and amyloid plaques being formed. So there are probably a couple different mechanisms by which diabetes could increase your risk for Alzheimer's disease. The question had to do with the kind of exercise, and does it need to be aerobic? So there are a couple of studies that suggest that aerobic exercise plus some strengthening exercise is probably best. Again, we don't know for sure, but that's certainly so. And I don't know about you, but when I take Pilates, I'm out of breath. That's aerobic. But a combination. But an exercise program where you're just stretching is probably not going to be nearly as effective as something that's aerobic. So in other words, getting your heart rate up, getting out of breath is going to be an important component. So what's the story with walking? So this is one of the challenges of an epidemiological study. We know that these individuals were reporting that they walked, but we don't know what else they were doing besides walking. Because I don't know about you, but the people I know who walk the most are also the ones who are the most physically active, the most intellectually active. So it may actually not have been the walking. We can't imply causality here, that walking in particular was associated with the reduced risk of dementia. But great question. What's the role of statins in maximizing brain health? So statins are a little controversial. There are plenty of studies out there that suggest that statin use is associated with better cognitive or brain outcomes. But that's not universal. And there are certainly clinical anecdotes of people reporting some decline in memory associated with statin use. So I think the whole issue of statins is fairly complicated. And so how do we explain these individuals who have lots of Alzheimer-related change, and I'll expand that to vascular changes or evidence for shearing effects from a brain injury? Well, how is it that these people are able to tolerate all that? The idea of cognitive reserve is a little on the abstract side, but it does try and capture the phenomenon of how some people, by virtue of starting off at a higher level, are going to be able to withstand an injury that someone else who's not as capable can't. But I don't think that tells the whole story. I really don't understand to a great degree how it is that some people are able to handle lots of negative brain changes. I think it's very intriguing. I think that as we grow to understand that better, we're going to hopefully be able to bottle that and have some interventions for the neurodegenerative diseases. So what effect does marijuana have on sleep and memory? Right. Yeah. And I actually don't know what impact marijuana has on the nature of the sleep and whether it impacts deep sleep, REM sleep. So I don't really know the answer to that. And there have been lots of debate in the literature about the long-term impact of marijuana use on memory functioning, with some reports saying that it's not so good, other reports saying that it's just fine. So I think the jury's still out on that. So the question is there a relationship between the number of neurons and cortical volume? There is one, but there are so many other things that influence cortical volume, including the size of the neurons, the size, the number of glial cells, and in fact, our brains grow tremendously when we're young, but we're not showing any increase in the number of our neurons. What we're showing is a huge increase in glial cells and myelination of those axons. So what are the brain changes in Alzheimer's disease? There is actually neuronal loss that's fairly significant. So are there gender differences in what, though?
In the causes, in the,
So the most amount of research that has tackled gender differences is around the use of hormone replacement therapy. And there's a lot of epidemiological evidence to support the idea that hormone replacement therapy or just having estrogen on board is actually protective for neurons, particularly in the hippocampus, and associated with better memory performance. But it's a very controversial topic, and I don't think that there's any definitive answers. And of course, there's interactions now, we think, between estrogen and some of these other factors that influence brain function. Way in the back. So the question is about the link between peripheral injury and what's going on in the brain. And so when there is a peripheral injury and peripheral inflammation, there is neural inflammation. There's a cascade of inflammatory cytokines and chemokines that occur in the brain. Exactly how that takes place or why and what's crossing the blood-brain barrier isn't clear, but it definitely does happen. And some people have actually used that to explain why after, if you like hurt yourself, you like burn your arm, you're going to feel tired and you're not going to really want to go out. That's sort of an adaptive mechanism that these neuroinflammatory processes are contributing to. So I think it's a normal part of the inflammatory cycle, and I think that the risk in aging is that this inflammation goes on for too long. It's kind of like stress. Stress is good. It's very adaptive. We need our stress mechanisms and fight or flight. It's a very, very important part of being alive. But you don't want to be stressed chronically for too long. Then it starts to cause multiple organ system failure. Great question. What about people who have chronic pain and are likely to have chronic inflammation? A lot of the studies with NSAID use were with people who had arthritis because they're much more likely to. So ironically, if people had arthritis would be at lower risk if they're taking the NSAIDs. The other groups that would be really important to study are patients with lupus, multiple sclerosis, and it's hard to separate out the direct effects of those inflammatory diseases because the diseases themselves have impact on the brain. And whether someone who has arthritis and is not taking any anti-inflammatories, whether they're at greater risk for Alzheimer's, I don't know the answer to that. It's a great question. Let's try. So the general consensus is that if you didn't get a good night's sleep, take a nap. Right. In the long run, it's better for your brain than getting it out. Okay, so the question is about and I get time for two more questions, and we wrap it up. I got my signal. The role of antidepressants. So that's a fairly complicated topic because then you have the effect of depression on the brain. And in older individuals who have depression, particularly late onset depression, a great many of us think that the brain is directly involved in the occurrence of those symptoms. So you've got the depression that's either a reflection of some early brain pathology or the possibility that the depression itself will manifest as things like reduced blood flow to the front part of your brain. So depression itself is not great for your brain. Depression is also associated with greater stress and poorer recovery from stress responses. So there are lots of reasons why depression is bad for your brain, and whether antidepressants have a positive effect on your brain by helping with the depression, and whether or not they have an independent negative or positive effect on the brain, I don't think is clearly understood, at least not by me. I don't know of any studies that suggest that caffeine use is either positively or negatively associated with risk of cognitive decline or dementia. So drink up. All right, thank you very much.